Hello and welcome to Bio40 Pre-Lecture 23. Today we're going to continue discussing cytogenetics. Let's pick up again with inversions. Inversions do not involve the loss or gain of genetic information, just a rearrangement of the order of the genes. Just like the other rearrangements we've discussed, inversions can be caused by improper repair of multiple double-strand breaks, or by crossovers between short homologous sequences. Because they do not involve a gain or loss of information, inversions rarely have phenotypic effects. The main exception is when the breakpoint of an inversion is in the middle of a gene. Both halves of the gene still exist on the chromosome, but not as a continuous sequence, so the gene will not be functional. Inversions can also affect fertility, which we'll get to in a moment. First, to understand how inversions behave in meiosis, we have to look at the two types of inversions. In paracentric inversions, the centromere is not part of the inverted region, but in paracentric inversions, the centromere is included in the inversion. Let's look at paracentric inversions first. When a normal chromosome and a chromosome with an inversion pair in prophase 1, one must form a loop to maximize the alignment of homologous regions. If a crossover occurs within the inverted region, it can be tricky to see what happens, but let's follow the strands. This chromatid is perfectly normal. This chromatid still has an inversion, but has all the normal regions it should have. But this chromatid goes A, B, C, centromere, D, A. It lacks the E region entirely, but has a duplication of A. Likewise, this strand progresses E, D, centromere, C, B, E. No A region, and a duplication of E. Here are these chromosomes after meiosis II. The recombinant gametes are unlikely to be viable. This means few, if any, recombinant progeny, and reduced fertility due to the lower percentage of viable gametes. Paracentric inversions have similar, but even more severe issues. Once again, a loop is formed during alignment in prophase 1. And again, the parental chromosomes are fine, though one of them still has an inversion. But let's look at the recombinants. This one goes centromere A, B, C, D, A, centromere. So that's a deletion of E and a duplication of A. But the biggest problem is the two centromeres. The final strand proceeds E, D, C, B, E. No centromere here. Now let's see what happens in anaphase. Our cells don't maintain DNA without a centromere, so the acentric fragment is lost. Meanwhile, the dicentric chromatid faces a major problem as the two centromeres are pulled in opposite directions. This will force a random break in the chromatid. Here we depict it occurring between regions A and D, but there is really no breakpoint that leads to a good result. These are the chromosomes at the end of meiosis II. So again, we're unlikely to get any recombinant progeny, and we'll see reduced fertility. However, there are research uses for inversions. When we want to suppress recombination in flies, we introduce balancer chromosomes, chromosomes with multiple inversions, thus making recombinant gametes non-viable. Now let's move on to translocations, where a portion of one chromosome becomes attached to a non-homologous chromosome. A reciprocal translocation is where parts of non-homologous chromosomes trade places. This too can be caused by multiple double-strand breaks or improper crossing over. Reciprocal translocations normally have little to no phenotypic effects because no genetic information is lost or gained. Though as with inversions, trouble can occur when the breaks disrupt genes. In fact, there is a very famous translocation with a major effect due to the genes it disrupts. The genes c-Abl on chromosome 9 and BCR on chromosome 22 typically code for normal benign proteins. But a reciprocal translocation can lead to a hybrid gene, one that starts with part of the BCR sequence and ends with a portion of the c-Abl sequence. The resulting BCR-Abl fusion protein causes uncontrolled cell growth, leading to cancer. Additionally, translocations can result in diminished fertility due to problems in meiosis. The issues in meiosis are only experienced by heterozygotes. When individuals are homozygous for translocations, the chromosomes can align without any trouble. But with heterozygotes, the only way for all the homologous regions to align is to form a group of 4 chromosomes. and meiosis cannot deal with this. Let's call the normal chromosomes N1 and N2, and the chromosomes with translocations T1 and T2. There are 3 ways the chromosomes could separate in anaphase 1. The 2 normal chromosomes can go in one direction, and the 2 translocations in another. This is called alternate segregation, since it's the alternate chromosomes in this depiction that move together. It's also possible that N1 and T2 could segregate together, while T1 and N2 do the same. Or T1 and N1 can end up together, as can N2 and T2. These latter 2 scenarios are called adjacent segregation. Of these, only alternate segregation leads to the formation of viable gametes. Let's look at the reason for this. With N1 and N2 together, the gamete has a full copy of each chromosome. And T1 and T2 also has all of regions A through L, just a little shuffled up. But when N1 and T2 are together, there's a duplication of D, E, F, and a loss of J, K, L, while the T1 and N2 chromosomes have a deletion of D, E, F, and a duplication of J, K, L. T1 with N1 and T2 with N2 gametes likewise have deletions of some regions and duplications of others. None of these last 4 gametes are likely to be viable. However, that last segregation is very uncommon. Normally the 2 blue centromeres will be pulled in different directions, as will the 2 brown centromeres. So given the common forms of segregation, half of meioses lead to viable offspring. This can cause semi-sterility. The kernels in this ear of corn are the progeny of an individual heterozygous for a translocation. Only half the gametes, those from alternate segregation, were viable. Another interesting aspect of translocation is the Robertsonian translocation. This is a reciprocal exchange between 2 acrocentric or telocentric chromosomes that leads to the formation of a large metacentric chromosome. Oftentimes, the tiny second chromosome resulting from such a translocation has little to no important information on it and can be lost. Events like this can cause problems, but they can also reshape a genome. Now, if we're discussing chromosomal rearrangements, we should at least briefly touch on transposons. These are pieces of DNA that exist just to jump from place to place within the genome. Transposons are the ultimate selfish genes. They don't benefit the organism they're found in. They exist solely because they're good at copying themselves. Transposons jumping into and disrupting pigment genes is why we sometimes see a speckled pattern in corn. There can be hundreds or even thousands of copies of a given transposon in a genome. This creates regions of homology in non-homologous chromosomes or on different locations in homologous chromosomes. Such regions make possible the crossover events that cause deletions, duplications, inversions, and translocations. Though researchers have also figured out how to use transposons as tools in biology research. Since we're discussing chromosomal abnormalities, we should revisit aneuploidy, the loss or gain of one or more chromosomes. This figure shows various types of aneuploidy, but the most common are monosomy, or the loss of a single chromosome, and trisomy, having one extra copy of a chromosome. Recall that aneuploidy is caused by non-disjunction errors during cell division. We mostly talked about this in meiosis, but non-disjunction can also occur in mitosis, leading to aneuploidy in somatic cells. When it comes to viable offspring, we talked before about how sex chromosome aneuploidies are more common than autosomal due to how dosage compensation with the X chromosome works. However, some autosomal aneuploidies are viable. Monosomy is always lethal, but there are 3 viable trisomies: trisomy 18, trisomy 13, and trisomy 21. Though while the first 2 are viable in the sense that they can make it to a live birth, they have very short life expectancies. The best studied trisomy is trisomy 21, or Down syndrome. Down syndrome is observed in about 0.1% of live births. The condition has a variety of different cognitive and physical phenotypes, though it has extremely variable expressivity, so it can present quite differently from individual to individual. About 1 to 2% of Down syndrome cases are familial, and we'll get into that in a moment, but the other 98% are sporadic. Of those cases, 90% are the result of non-disjunction during gamete formation in the mother, usually in meiosis 1. In this table, you can see how the chances of having a child with Down syndrome increases with the age of the mother. So what is it that makes non-disjunction more likely over time? The answer has to do with the timeline of gamete development. In women, gamete development begins before they're even born, but the process is halted at prophase 1 and does not continue until ovulation. So the cells stay arrested in the middle of meiosis for decades, and the longer this lasts, the higher the likelihood of non-disjunction. Now what about those 1 to 2% of cases that are familial? These are the result of Robertsonian translocations between chromosomes 14 and 21. The translocation by itself is not a problem. Little information is lost from the small ends of these chromosomes. So an individual with a translocation and one normal copy of chromosomes 14 and 21 will be just fine. As we've often seen to be the case, the problem occurs in meiosis. There are 3 ways the chromosomes can segregate in meiosis. If the chromosome with the translocation goes to one gamete, and the normal chromosomes go to the other, all offspring will have a normal phenotype. But if the translocation and chromosome 21 segregate together, that gamete will give rise to a child with Down syndrome, while the other will have monosomy 21, which is not viable. Likewise, if normal chromosome 14 segregates with the translocation, leaving normal chromosome 21 by itself, the offspring will have trisomy 14 or monosomy 14, neither of which are viable. Out of 6 possible offspring, 1 has a normal set of chromosomes, 1 carries a Robertsonian translocation but is phenotypically normal, 1 has Down syndrome, and the remaining 3 are non-viable. Let's close out by looking once more at the relationship between chromosomal rearrangements and evolution. Closely related species tend to have very similar arrangements of chromosomes. If you compare our chromosome 1 to that of our closest ape relatives, you see they line up quite well. Though our chromosome 2 lines up with 2 different ape chromosomes, supporting the idea of a fusion that occurred in our species past. The more distant the relatives you compare, the more rearrangements you see. This raises the question, are rearrangements simply something that occurs as we evolve, or are they a driving force of that evolution? And that wraps up another topic. Next time, we shift gears again and discuss bacterial genetics.